Hi, my name is Kayley Besbris, and this is my final presentation for Anatomy and Physiology 1 with Dr. Marks. Our final presentation was based on how the plasma membrane transport mechanisms, sodium-potassium pumps, action potentials, and the autonomic control apply to our digestive systems and how we absorb nutrients. Now, for our plasma membrane transport, the nutrients are absorbed into the small intestine through the various transport mechanisms across our plasma membranes of the epithelial cells lining our small intestines. The mechanisms of transport include simple diffusion, facilitated diffusion, and the active transport. The small intestine's structure has a large surface area with specialized cells helping facilitate absorption. The plasma membranes of many epithelial cells are divided into distinct apical and basolateral domains that differ in the function and protein composition. For example, epithelial cells of the small intestine function to absorb nutrients from the digestive tract. The apical surface of these cells, which face the intestinal lumen, is therefore covered by the microvilli and specialized for nutrient absorption. Our basolateral surface, which faces the underlying tissue and the blood supply, is specialized to mediate the transfer of the absorbed nutrients into our circulation. Now, we have simple diffusion. In the small intestine, the simple diffusion is a key process of nutrient absorption. This is particularly for smaller molecules like our fatty acids, our glycerol, and some water-soluble vitamins. This process relies on the concentration gradient, where nutrients move from an area of higher concentration, like our small intestines, lumen, to an area of lower concentration, like our bloodstream. The process of simple diffusion takes place beginning with digestion and nutrient release. This is where digestion will break down large foods into smaller molecules like fatty acids, glycerol, and monosaccharides. Then we have the formation of micelles, or for fats. This is where our fatty acids and monoglycerides combine with bile salts to form the micelles, which are more water-soluble structures that can transport these fats into our intestinal linings. Then we have the movement across the membranes, and this is where the nutrients, where they're dissolved in water, move across the intestinal lining, specifically in the villi and the microvilli, through simple diffusion, meaning they move down our concentration gradient without the need for energy or for protein transporters. And then we have the absorption into the bloodstream. The absorbed nutrients then enter the bloodstream through capillaries within the villi, where they are ready to be transported throughout our bodies. And then lastly, we have the lipid reassembly inside the intestinal cells, fatty acids, and monoglycerides. This is where they're reassembled into triglycerides and packaged into chylomicrons, where they're then transported to the lymph systems. Then we have our facilitated diffusion. Facilitated diffusion helps nutrients across the cell membrane by using our carrier proteins, moving them from an area of high concentration to an area of low concentration. And then we have, after our food is digested, which results in the nutrients like fructose, need to be absorbed into the bloodstream used by our bodies. Then the facilitated diffusion relies on the specialized carrier proteins embedded in our cell membrane. These proteins act as doorways that allow our specific nutrients to pass through. Lastly, we have the active transport. And the active transport moves the nutrients across the concentration gradient, requiring energy. This is crucial for absorbing some nutrients like glucose and amino acids, which are transported into our intestinal cells, which, with the help of the proteins like SGLT1, sodium glucose cotransporter 1, and GLUT2, glucose transporter 2. The active transport is a cellular process that moves across the cell membrane against their concentration gradient, which means from an area of lower concentration to an area of higher concentration. This process requires energy, typically in the form of ATP, to fuel the movement. In our small intestine, the active transport is essential for absorbing nutrients like glucose, amino acids, and some vitamins, which are needed by the body, but might be lower concentrations in the intestinal lumen than inside the cells. And then we have the sodium glucose cotransporter 1. This protein, located on the apical membrane of the intestinal cells, is responsible for the secondary active transport of glucose and galactose. It works by coupling the movement of sodium ions down their concentration gradient into the cells with the movement of glucose or galactose against the concentration gradients. Then we have the GLUT2 transporter. This protein is found in our basolateral surface, or the blood facing, and this membrane facilitates the transport of glucose and galactose out of the intestinal cells and into our bloodstream. The two mechanisms of the active transport are primary and secondary active transport. Primary active transport involves a protein pump that directly uses ATP to move the substance against the gradient. An example is the sodium-potassium pump, which maintains the sodium and potassium gradients necessary for other transport processes. The secondary active transport utilizes the electrochemical gradient established by primary active transport. This is an example of movement of sodium down its concentration gradient, where it is coupled with the movement of another molecule like glucose against its concentration gradient. Then we have the sodium-potassium pumps. The sodium gradient established by the sodium-potassium pumps provide a source of energy that is frequently used to power the active transport of sugars, amino acids, and ions. The epithelial cells lining the intestine provide a good example of the transport driven by the sodium gradient. These cells use active transport systems in the apical domains of their plasma membranes to take up dietary sugars and amino acids from the lumen of our intestines. The uptake of glucose, for example, is carried out by the transporter that coordinately transports the two sodium and the one glucose into the cell. The flow of the sodium down its electrochemical gradient and provides the energy required to take up the dietary glucose and to accumulate high intracellular glucose concentrations. Glucose is then released into the underlying connective tissue at the basolateral surface of the intestinal epithelium, where this is transported down the concentration gradient by facilitated diffusion. Then the uptake of glucose from the intestinal lumen and its release into the circulation. And then we have our action potentials. This is where several types of nerves work together to regulate digestion, both directly and indirectly. And this includes our intrinsic nerves, which connect digestive organs to the brain and spinal cord, forming a crucial link of gut-brain connection. These nerves belong to our autonomic nervous system, which controls involuntary functions like digestion. Then we have the vagus nerve. This is a major parasympathetic nerve involved in charge of regulating muscle contractions, glandular secretions, and the movement of food through the digestive tract. We also have the sympathetic nervous system, which slows down digestion by relaxing the stomach and the intestinal muscles and decreasing blood flow to the digestive organs. We also have the intrinsic nerves and in the enteric nervous system. This is a complex neural network embedded within the walls of the gastrointestinal tract, and with the ability to function independently from the brain and the spinal cord, controlling various aspects of our GI physiologically, like motility, secretion, and blood flow. This is often referred to as our second brain because of the autonomy and the complexity of it. The enteric nervous system consists of these neurons: the intrinsic motor, the intrinsic interneurons, and the sensory neurons. The intrinsic motor fibers regulate our muscle contractions for mobility. The intrinsic interneurons communicate with our enteric nervous system, and our sensory neurons detect stimuli like stretching or chemical gradients within our GI tract. And then lastly, we have the autonomic control. This is our autonomic nervous system, which regulates digestion through its two branches, the sympathetic and the parasympathetic nervous systems. The parasympathetic nervous system promotes digestion by increasing intestinal mobility, stimulating digestive secretions, and relaxing our sphincters to allow food to move through the digestive tract. This consists of the vagus nerve, which plays a crucial role regulating the digestive the digestion by stimulating motility in the small intestine and our colon, as well as controlling our gastric motility and secretions. We also have increased motility secretions to promote the movement of food through the digestive tract, and this also stimulates the release of digestive juices and enzymes. We have the sympathetic nervous system, which is in charge of inhibition of digestion, which reduce mobility of secretions, and decreased rate of contractions in the digestive tract, which also constricts the sphincters, which can slow down or even stop the movement of food. The enteric nervous system helps to regulate digestion by controlling muscle contractions, secretions of digestive juices, and our blood flow to the digestive organs. The sympathetic and parasympathetic and enteric nervous systems work together to help coordinate the complex process of digestion, the absorption, and the elimination of waste in our digestive tracts. And then lastly, I have the references that I used to gain all of my information. Thank you.